I'm Lisa Chardon. Welcome to the Aging Mind Foundation Lecture Series, sponsored by Belmont Village Senior Living. Today, we have Dr. Michael Rugg, Director of the Center for Vital Longevity at the University of Texas, Dallas, as our speaker. We're going to hear from Mercedes Carr, President of Belmont Village Senior Living, and then we'll hear from Larry Hulshoff, founder of the Aging Mind Foundation. She's going to provide an overview of our mission and impact in finding a cure for Alzheimer's disease and other dementias.
Hello everyone, I'm Mercedes Carr, President of Belmont Village Senior Living. I'm pleased to introduce this installment of the Alzheimer's Research Webinar Series with the Aging Mind Foundation. For almost 25 years, Belmont Village has been home to so many interesting, accomplished, and beloved residents, many of whom suffer from dementia or Alzheimer's disease. We strive to create purposeful, engaging, and joyous experiences for each of them and know that our evidence-based therapeutic programs help them to live better, wellness-inspired lives. We're always learning and continuously innovating to serve our residents' needs, so I look forward to this series. But before I turn it over to our speaker, I want to reiterate Belmont Village's commitment to those who are navigating this complex and difficult condition and to remind you that we're always here to help. I hope you enjoyed today's discussion.
As the founder of the Aging Mind Foundation, it is my great privilege to welcome you today to our 2021 Aging Mind Foundation Lecture Series. These lectures are generously sponsored by Belmont Village Senior Living. Our mission at the Aging Mind Foundation is to raise money to fund scientific research that seeks the cause of Alzheimer's disease and other types of dementia. Since 2014, we have raised over $4 million, providing research grants to the top minds in neuroscience. The short following video reflects some of the progress and impact the Aging Mind Foundation has made in years past. Our presenters will discuss their latest advancements in their research, followed by a brief question and answer session. Please submit your questions via Zoom, and they will be answered in the time allowed. Thank you so very much for your support of the Aging Mind Foundation. And by the way, I am certain you will enjoy today's lecture.
Dementia is a progressive brain disease that slowly and relentlessly steals a person's memory and ultimately their identity and ability to function. Alzheimer's disease is the most common form of dementia, and despite its general public image, it affects more than just the elderly. It also greatly impacts their caregivers and loved ones and is the costliest disease in the country. With 6 million Americans living with Alzheimer's, the estimated cost is $355 billion per year. And as our growing population ages, cases and costs are expected to triple by 2050. It's also increasingly impacting a younger demographic as early onset Alzheimer's, diagnosed between 30 to 65 years of age, becomes more common. Dementia is a horrific and cruel disease that steals your memories, your ability to function, and erases a life lived. 1 in every 3 seniors dies with Alzheimer's disease or another form of dementia, and 2 out of 3 people diagnosed are women. Will you be one of those statistics? Will your partner? Your children? In the United States' top 10 most deadly diseases, Alzheimer's is the only one that cannot be prevented, treated, or cured. And while deaths from diseases like HIV, heart disease, and cancer have steadily declined, deaths from Alzheimer's have increased. It's time that Alzheimer's disease and other types of dementia begin to get the funding and attention they deserve. It's time we change the course of Alzheimer's disease. Since 2014, Aging Mind Foundation has been continually committed to raising funds for vital brain research. We're focused on funding research that seeks the cause of Alzheimer's disease and other types of dementia, because we must understand the root cause in order to develop treatments and prevention. Partner with us, join us, donate. Help fund the necessary brain research that will alter the course of Alzheimer's and dementia, so future generations will not have to suffer the harsh realities and cruelty of losing loved ones to this disease. Let's make Alzheimer's the only memory we forget.
Now I would like to introduce Dr. Michael Rugg. As I said earlier, he is the director of the Center for Vital Longevity at the University of Texas, Dallas. His talk today is titled, "Why Do Our Memories Get Worse as We Age? Insights from the Study of Neural Selectivity." As we hear from Dr. Rugg today, please submit any questions to the Q&A feature, and we will read them to Dr. Rugg after his presentation. I'm thrilled to turn it over to Dr. Michael Rugg. Good morning, Michael.
Hi, Lisa. Thanks so much for the introduction. And I'm going to share my screen and get cracking on my presentation. Good morning, everybody. I apologize at the outset for the rather cheating nature of my title, "Why Do Our Memories Get Worse as We Age?" The short answer to that is we know far less about that than we need to know. And in fact, if it wasn't for that, I would be out of a job. What I'm going to do today is talk a little bit about some of our work that has looked at brain imaging and how that's given us insights into memory performance and age-related memory problems. And I'm going to end up by talking about a new line of research in our laboratory, which we think is very exciting and which we hope will give us some additional insights. So just before I start, let me acknowledge the support for this research, National Institute on Aging, the National Science Foundation, and the Aging Mind Foundation, whose support we are extremely grateful for. I also need to say at the outset, I have no competing interests in any of the research I'm going to be talking about. So I should also start by acknowledging people in my lab who took the lead on the work I'm going to be describing today. It takes a village to do this kind of research. In some ways, I'm just the spokesperson for a group of extremely talented people who do the actual work. Josh Cohen was Aging Mind Foundation postdoctoral fellow in my lab for about 15 months. He headed up some of this work. Marianne de Chastelain, who's a long-standing postdoctoral researcher in the lab also. Paul Hill, who was a postdoc in my lab, is now working at University of Arizona, also headed up the work, and Sabina Strakova, who's a fourth-year graduate student in my lab, also has played a major role in the work I'm going to be talking about today. So let's start off with some questions. I am going to be talking primarily today about how memory declines in, as far as we can tell, cognitively healthy people, people who we select very carefully not to be apparently at risk for Alzheimer's disease at the time that we're researching them. And it's a very reasonable question to ask why, if we're so interested in Alzheimer's disease, would we be studying healthy people? And I will just give three practical replies to that perfectly reasonable question. The first is that we will not understand brain diseases, and this includes Alzheimer's disease, if we don't know how healthy brains work. The study of normal function and the study of how things go wrong in disease are essentially two sides of the same coin. And just by way of an example, the great majority of the work that has gone into understanding cancer and has led to spectacular increases in life expectancy for people suffering from cancer has not been work on cancer directly, but has been work on trying to understand how normal cells replicate and how they prevent errors of replication. And by the same token, if we want to understand how things go wrong in diseases like Alzheimer's disease and similar dementias, we really do need to understand how the brain works when it's going right. Second, even among people who don't have Alzheimer's disease or don't seem to have it, age-related memory decline can be very severe. It can be so severe it interferes with everyday life. We would like to know what factors are responsible for these differences in later life memory abilities. And in particular, we'd like to know whether any of them are related to the pathology that is seen in Alzheimer's disease, and indeed whether or not any of these later life memory difficulties are predictive of the even more severe memory impairments that accompany Alzheimer's disease. And finally, by studying people who at the time that we study them are, as far as we can tell, healthy, and then following them over time, we can identify differences in the brain's function and structure that are very subtle and that we may not see if we don't go looking for them, but which are linked to later cognitive decline and perhaps are predictive of those people who are likely to succumb to Alzheimer's in later life. This is a particularly important issue because it seems pretty clear that if we want to prevent or to halt Alzheimer's in its tracks, we're going to have to intervene very early in the course of the disease, and ideally before people are showing any overt symptoms of the disease at all. So let's just spend a moment to look at memory across the healthy lifespan. This figure here demonstrates memory performance on the y-axis here as a function of age. And it does so for 4 different kinds of memory. One of the most important insights in the psychology of memory and the neuropsychology of memory over the last 50 years or so has been the realization that memory is not a single cognitive faculty that is supported by a single set of brain regions. Rather, memory is an aggregate of different kinds of memory functions that have at least partially distinct neurological underpinnings. Episodic memory is what we refer to as memory in everyday life. This is the kind of memory that allows you, for example, to know what you had for breakfast yesterday, what you bought in the supermarket a week ago, where you parked your car this morning, etc. So episodic memory is our memory for unique everyday events. And you can see here that episodic memory declines pretty sharply and with an accelerating function over the lifespan. By contrast, semantic memory is essentially memory for world knowledge. For example, knowing that Paris is the capital of France would be an example of semantic memory. And as you can see, over the same period that episodic memory is beginning to decline, semantic memory continues to increase over the lifespan. And in fact, in my opinion, this drop-off here is rather overestimated. World knowledge and semantic memory holds up pretty well across much of the lifespan. Short-term memory is hardly affected at all. This is the kind of memory, if you can remember those days, that you need to look up a phone number in a phone book and hold on to it while you rather charmingly dial the number. For the younger people in the audience, that example probably goes nowhere. But anyway, this short-term memory, just holding information in mind for a few seconds, is very little impacted. And the same is true for what here is called priming. This is a whole collection of different kinds of memories that have in common that they get better with practice. And for example, the ability to speed up and to do a skill more and more rapidly over time is an example of priming. And again, this is very little affected by healthy aging. So we are going to focus on episodic memory because that is the form of memory that is most vulnerable to healthy aging. And it is also the kind of memory that is most heavily affected and affected most early in people who are developing Alzheimer's disease. This figure here is some data from our lab. Every one of these little diamonds here is an individual participant. And we're looking here at groups of people in their 20s, groups of people in their 40s and 50s, and people aged, roughly speaking, between 65 and 75. And these are the typical sorts of age ranges that we study in my lab, in many of our studies. I should say that in our most recent work, we're extending this age range now out to 90 and 90 plus. But at the moment, most of the data that we have is for people aged, roughly speaking, between 65 and 75. And what we're looking at here is performance on an extremely sensitive test of episodic memory. This is the average performance of our three groups. And this shows the typical age-related differences that one sees in episodic memory. But notice that there is huge variability within these age groups here. And in fact, quite a few of our older people are performing above the average, the mean of our younger group. And there are plenty of younger individuals here who are not performing as well as our older individuals. And what we're interested in trying to understand is what is it that explains these large individual differences in memory performance that we see, not only in older people, but across the lifespan. And of particular interest is what can we do, if anything, that would allow people down here to be more like people up here. So those are the sorts of issues that we've been looking at. So I am going to talk today exclusively about research that we've done using a technique called functional magnetic resonance imaging, or fMRI. And this is not because other methods are not important, but in a short talk, I'm going to focus just on this one aspect of our research. This is an MRI scanner. People lie on this bed with their head in this head coil, and they go inside this tunnel here. And the MRI system, as I'm sure many people are familiar, allows us to get exquisite, highly detailed pictures of the brain's anatomy. So this is looking at the brain as if we were looking through the nose, and we're looking at a sort of salami slice of the brain here. And this is looking at the surface of the left cerebral hemisphere, reconstructed from an MRI scan. Work in our lab and in other labs in the CVL, much of that work is concerned with analyzing these brain structural images and trying to understand links between differences in brain structure and differences, for example, in memory function. But that's not what I'm going to be talking about today. We can also use MRI to look at brain function. And there are a number of methods for doing this, but essentially what we can do is tune the scanner so that it picks up differences in blood oxygenation, which are directly related to differences in the activity of the underlying brain regions, the underlying neural regions, the neurons in the brain. And what we're looking at here, for example, are regions where we see more activity when people succeed in remembering a prior event that we've had them learn in the laboratory than when they forget that event, when they fail to remember it. And of particular importance, this region here, shown on both the left and the right side of the brain, is called the hippocampus. And the hippocampus is a brain region which is critically important for episodic memory function, and which also is highly vulnerable and is impaired early on in the course of Alzheimer's disease. I'll return to this in a moment. But the bottom line is that by using functional magnetic resonance imaging, fMRI, we can get insight not in just to the brain structure, but to its function. So we can ask questions like, can we find functional correlates of memory performance? Do we find differences in brain function as a function of age that explain differences in memory in older and younger people? And we can ask these sorts of questions. And that's what I'm going to briefly present to you today, some results along those lines. So when we are studying memory, we draw a distinction between essentially three different stages of memory function. You can think of this rather simplistically as being like the library model of memory. For memory to work well, information has to get encoded into memory. That means that when you are experiencing events in the world, those events are somehow represented in the brain and transformed into a durable memory trace. That memory trace has to be stored, and then later it has to be retrieved. And all three functions here have to be working normally if we're going to have a normally working memory function. And in work I am going to be talking about today, going to be talking a little bit about what goes on at the time of encoding information into the brain. That predicts whether we'll remember it later. And I'm going to say a little bit also about what happens at the time of retrieval, things that go on in the brain that are important for us to recover and represent an accurate representation of a prior event that we've experienced. So let me just start with some relatively old work, but work which we think is still important and still gives us important insights into some of the factors that determine successful memory performance in older people. So we're looking here at an unfolded picture of the left cerebral hemisphere of the brain. And the oval here is in a region of the brain called the left inferior frontal cortex, which is extremely important for our ability to encode complex information into memory. And what we see here is that this region of the brain is more active when people succeed in remembering and succeed in encoding an event. That means they're looking or processing an event that they later remember, compared to when they're processing an event that they later forget. So this brain activity here is predicting in people who are lying in the scanner whether events that they're experiencing are going to be remembered later or not. And what we find in this brain region and on the other side of the brain also is that this effect exists in young, middle-aged and older people. So this bar chart here tells us how much more active this region is for events that we're going to remember than events that we're going to forget. And we can see that in younger and middle-aged people, we get more activity in the left frontal cortex, but we see absolutely nothing in the right frontal cortex, the matching region on the other side of the brain. But in our older people here, we again see activation in the left inferior frontal cortex, but that is accompanied by activity in the right hemisphere as well. Moreover, only in older people do we find a relationship between the size of this activity and how good your memory is. So in older people, the more that you can turn this region on, the more likely you are to remember information, and moreover, the better is your memory. But we only find this in older people, and we find this activation on both sides of the brain in older people than younger people. And this makes us think that what we're seeing here is evidence of a region of the brain that is particularly vulnerable to increasing age. And ultimately, by the time that you become a relatively elderly person, the vulnerability of this region of the brain is exposed. And what we're seeing here is the results of people who have a relatively highly functioning compared to a relatively low functioning frontal cortex. So we call this what we would call a bottleneck region. As you grow older and this region degrades in its function, it becomes more and more of a bottleneck that helps to control how successfully you will be able to remember information. Switching quickly to retrieval, this is from the same participants we've just been looking at, but now we're looking at regions of the brain that are more active when you succeed in retrieving information than when you forget that information. And here is our old friend, the hippocampus. And what we find in this case is that regardless of age, the more you can turn on your hippocampus when you're retrieving information, the better is your memory. In other words, it doesn't matter whether you're 20 years old or 70 years old, your hippocampus plays an important role in memory function, and that role seems to be very similar. So unlike the frontal cortex, the hippocampus is not a bottleneck region. It doesn't matter what your age is. You're going to do better as a memorizer if you can turn this region on more at the time that you're trying to remember information. One interesting result that I just briefly want to mention is that the older people in this study here, who we scanned along with our younger and middle-aged people, we brought them back and we retested their memories 3 years later. And we discovered that in the left hippocampus here, the extent to which you could turn this region on predicted how good your memory was 3 years later. So what we're looking at here is on the y-axis here is how much your memory declined over time. As this number goes up, you're someone whose memory suffered more over the 3 years than someone down here. And what you can see is the more that you were able to turn on your hippocampus 3 years ago, the less is your memory declining over that 3-year period. And we're following this up now in a much larger scale study. And we obviously also want to ask whether or not this effect is predictive, not just of memory decline, but of pathological memory decline, for example, in people who go on to develop the early stages of Alzheimer's. The last thing I will talk about briefly is this concept of neural selectivity. So it turns out, and this is one of the remarkable discoveries from the use of these functional imaging methods in people over the last 30 years or so, that the visual areas of the brain, and what we're looking at here is the underneath of the brain, the underside of the brain. We've taken the cerebellum off, and we're looking at the posterior regions of the brain, the so-called ventral aspect of the cerebral cortex. It turns out that these brain regions are organized in a material selective way. So different regions of the brain are selectively activated, for example, when we read, when we recognize faces, when we recognize objects, and when we are processing spatial information such as scenes or complex three-dimensional spatial entities. These different regions have wonderful jargonized names, which I'll introduce in a moment. But the bottom line is they are evidence of what one would call selectivity in brain function. And for both theoretical reasons and also empirical experimental evidence shows that there is a reduction in how selectively these brain regions respond to their so-called preferred stimuli as we grow older. And some of the early work showing this relationship between a decline in selectivity and age was actually conducted by my dear friend and the founding director of the CVL, Denise Park. So I'm going to be talking about some of the work that we've been doing on neural selectivity and say why we think this is relevant to understanding age-related memory differences and also relevant potentially to understanding what may be happening in the very early stages of Alzheimer's. So here are real data. What we're looking at here is a real brain, and we're looking at 48 participants, the average data from 48 participants. These yellow regions here are regions that are more selective. They respond more vigorously when people are looking at scenes than when they're looking at objects. I apologize to cat lovers for lumping cats together with objects, but as far as the brain is concerned, at least for our experiment, a cat is an object. So these yellow regions here are responding selectively to scenes relative to objects. And this region goes by the glorious name of the parahippocampal place area or parahippocampal cortex. These red areas here are regions in the occipital cortex on both sides of the brain that respond more vigorously to objects than they do to scenes. So what we're looking at here is exactly what I showed you in cartoon form, but this is in real brains. We can separate out regions that respond selectively to scenes and regions that respond selectively to objects. And in this experiment, we had people, older and younger people, lying in the scanner, and they were asked to make ratings about objects and scenes while we measured their brain activity. And we used a metric. I'm not going to describe it right now. I'm happy to do so if anyone would like to know the details. But we used a metric that basically measures how much more selective, for example, this region is when it's seeing an object than when it's seeing a scene. And what you can see here is that regardless of whether you're an older or a younger person, your LOC, so-called, responds selectively to scenes compared to objects, but it doesn't do so to any greater extent in younger people than older people. And we can compare that with this striking result here. Younger people show considerably more selective response to scenes than they do to objects than older people do. Really striking result. Moreover, when we ask about the relationship between selectivity and memory performance, we find that regardless of age, doesn't matter if you're a young person or an older person, the more selectively that this region responds to scenes compared to objects, the better is your memory for these items. And it doesn't matter whether it's memory for scenes or for objects, it's irrelevant. So this region here, the selectivity of this region here, appears to be correlated with and predictive of your ability to remember this information when we test your memory for it some 20 to 30 minutes later. And just to reiterate, whether you're an older person or a younger person, the more selectively that you encode information in this region, the better you remember it later. This is another experiment which is trying to tackle the same sorts of things. In this experiment, we again have our old friend, the parahippocampal place area. We have another scene selective area, which I'm just simply going to refer to as RSC. It's actually retrosplenial cortex, but that's a bit of a mouthful. And then in this experiment, rather than using objects, our other class of stimuli were faces. And we looked at selective activity in two face selective regions, the so-called fusiform face area in red and the occipital face area in purple. We don't need to care about the names. We just need to know that we're looking at face selective and face selective and scene selective brain regions. And we're presenting the data a little bit differently here. Again, we're still looking at a measure of selectivity, but now we're showing every individual participant. And these black dots are the average responses of our younger and our older groups. So we're looking here at the average selectivity in these two face selective regions for older and younger people. And you don't need to use statistics to know that there is no difference here. So just as is the case for objects, in the cases of faces, older and younger people appear to represent these faces with equally high levels of selectivity. When we move to scenes, things could not be more different. So now, just as before, we find that younger people, their scene selective regions, both the PPA and the PPA and the RSC, are much more selective. They respond more vigorously to scenes than to faces than is the case in our older people. Same results as we saw before, but now with faces instead of objects as the alternative category. So we're replicating our prior result. Moreover, just as was the case previously, and I should emphasize, these are completely separate samples of participants. There's no overlap between these two experiments. Just as previously, we find that the degree of selectivity in the PPA, the parahippocampal place area, is predictive of memory for faces and scenes. And again, this is independent of age. It doesn't matter if you're young or if you're old. So once again, we get convergent evidence or replicating evidence that there's something special about this region that predicts memory function. And it doesn't just predict memory function for the region, for the information it's interested in. It predicts memory for apparently all sorts of categories. And then just finally, the experiment I've just described to you was done with people lying in the scanner, both when they were processing scenes and faces, and later when we asked them to remember them. And the way we did this was as follows. So this is the condition that I've just shown you. People are lying in the scanner. They saw the scenes, but they were paired with a word. and the stimuli could be paired either a scene and a word or a scene and a face. People only saw one word pairing. So if you saw raccoon with a scene, you wouldn't see raccoon with a face, but this was the basic idea. So people are lying in the scanner, and what they were asked to do was to form associations between these words and these faces, or words and scenes. And that's the data I've just shown you. We kept them in the scanner. Oh, I should say, and this is the regions that I've just described to you already. We kept people in the scanner and we tested their memory for these events. And the way we did that was that they only saw the words. So you lay in the scanner and you did this task, and then we keep you in the scanner. We show you these words, and your job is to say, did this word get paired with a face or did it get paired with a scene? And that's your memory task. That's what you have to do. And the idea here, which is supported by an enormous amount of evidence, is that when you succeed in remembering a specific class of event, for example, a face, you what is called reinstate or reactivate the pattern of brain activity that existed when you originally experienced that event. So the idea is, to remember a face, you have to turn back on regions of the brain that were sensitive when you were presented with that face in the first place. And in the jargon of memory, this is called reinstatement. The idea is that the hippocampus drives a reinstatement of the original event in the brain regions that originally encoded that event. So that's the basic idea. What that means is that we can measure selectivity when people are retrieving information in exactly the same way that we can measure selectivity when they're actually experiencing that information. So our question here is, to what degree do we see differences in selectivity between older and younger people when they are remembering this information that mirror the differences that we saw when they were experiencing the information in the first place? And these are the data. So this is selectivity at retrieval now, shown on the y-axis. And again, we have our younger and our older people. And we're looking at the extent to which face selective cortical regions are more selective for faces than they are for scenes. And remember, nothing is being presented to our participants here. This is literally a memory effect. All they're seeing on the screen is a word. And you can see that we get rather meager, but statistically significant selective reinstatement in both older and younger people. But this difference is nowhere near statistically significant. By contrast, when we have people remembering scenes, and we look in scene selective cortex, we see an exact analog of what we saw when people were actually experiencing these stimuli. In other words, we recapitulate the differences in neural selectivity for scenes in the same brain regions when people are retrieving these scenes as when they are experiencing them. And we know from experiments that have been conducted both in my lab and in other people's labs that the greater selectivity with which you can retrieve this information, the more detailed and the more vivid is the memory. So the bottom line, the assumption here is that our younger people are retrieving this information with more vividness and more precision, if you like, than our younger people are, than our older people are. Don't worry about the complexity of this figure. I apologize, but the only thing you need to know is that this straight line here is representing the relationship between how selectively you retrieve information and how selectively you represented that information when you originally perceived it, when you were experiencing it. So the horizontal axis here is how selectively, for example, you were representing a scene in your parahippocampal place area when you were experiencing that word scene pairing. And this is how selectively you can represent that scene when you remember it. And the bottom line is that, as you can see, this correlation is very strong. And in fact, if we take into account selectivity at the time that people are experiencing this information, this difference goes away. So this difference is entirely due to not what's going on at retrieval, but what's going on at encoding, what's going on at the time that you're actually experiencing the information. And this has led us to a somewhat tongue-in-cheek principle, garbage in, garbage out. The basic idea is you can only remember information as well as that information was represented in your brain at the time you experienced it. If you have low selectivity and low precision in the way that you represent information initially, you will have low precision when you remember that information later. So just to quickly summarize, I hope if I haven't made it clear, let me just be quite explicit. There is no single cause of age-related memory decline. Different factors may be important in different people, and it is a big challenge to figure out which factors are most important in different people that underlie their memory decline. Having said that, we believe that one important factor is the selectivity with which events are represented in the brain as they're experienced. And this is especially so, we believe, for the parahippocampal cortex, the so-called parahippocampal place area, where selectivity is reduced with increasing age and where it also predicts memory and performance. We think that there may be no accident as to why the parahippocampal cortex is so selective to age, and there may be a clue here as to why it predicts memory performance. So this is the inside of the right hemisphere of a brain. This is the hippocampus here. The parahippocampal cortex sits just here, just underneath and to the side of the hippocampus. And in front of it are other cortical regions, which are also very important for memory. The critical point is that the earliest degeneration that occurs in Alzheimer's disease and is associated with the accumulation of the tau protein and with atrophy of the brain starts here. So in most cases of Alzheimer's disease, and the earliest detectable pathology, and this pathology can be detected in people who are pre-symptomatic and even preclinical, they're not showing particularly significant memory problems at all. The pathology starts here and it rapidly spreads through the cortex into the parahippocampal cortex. And our current hypothesis is that the unique sensitivity of the parahippocampal cortex to this combination of aging and memory performance may be telling us something about its sensitivity to Alzheimer's-like pathology. And that's something that we're following up right now in subsequent work. And then finally, selectivity during the encoding events predicts the precision with which the events are later remembered. This is the garbage in, garbage out principle. And this leads to the idea that if we could find some intervention that improved how precisely or how selectively we could represent events at the time we were experiencing them, we would improve people's memory down the line. And that is an exciting possibility, which a number of labs are also thinking about. So that is the end of my presentation. I thank you very much for your attention.
Thanks, Dr. Rugg. It was a great presentation, and we have some questions that have been submitted from the audience. The first question is, how did you gather the people or the sample for the study? Are they all affiliated with the university?
No. So we, particularly for our middle-aged and our older individuals, we advertise in the community. The majority of our older and our middle-aged people are people who see our advertisements and contact us and volunteer. With our younger participants, a higher proportion than we would like are recruited from the student population of UT Dallas. We've had a great deal of difficulty actually in getting younger people from the more general population, for not least because if they're often working. But no, particularly with our older people, these are people, community dwelling individuals.
And the second half of that question is, are you looking for any particular level of education when you're looking for participants?
So no, we're not. What we do is, for the kinds of work I've just been describing, we do give people a cognitive test battery. And what we're doing is we want to make sure that the people we're studying are unlikely to be on the cusp of or at high risk for cognitive decline. We don't care about education, but if people score significantly below the average level for their age, they are not eligible for these studies. Right now, we're running a study where that is not the case. And we are very happy to take people right across the spectrum.
Okay. The next question is the use of alcohol or other substances, medical or recreational, considered a disqualifier when it comes to research?
No. So again, we screen. We would be unhappy about taking someone who reported that they had a history of substance abuse. That means very high levels and regular use of something that's interfering with their everyday lives. But we have no disqualification with respect to what one would call, normal use. I guess if I count myself as a social drinker, I would. You may differ, Lisa, but I would consider myself to qualify for one of my own experiments.
Okay, great. The next question is, are there any brain exercises that can turn on your hippocampus?
So that's a great question. That is part of a larger question, essentially, of are there things that you can do that maintain the efficiency and the functionality of these regions that are critical for memory? I focused on hippocampus. The hippocampus doesn't work in isolation. There is an entire neural network that has to function successfully for memory to be successful. Yes, so people have addressed this question for a long time, and the answer is the jury is out. We really don't know, frankly. Having said that, there is data from more than one study that suggests that physical exercise does seem to have an impact on the structure of the hippocampus and to improve its function, to improve its structural integrity. And there are tantalizing suggestions that might translate into better memory performance. But I would put those data on the very much on the tentative side of things.
Okay, great. Does association between objects and scenes help people maintain the memory or memories longer?
Yes. So one of the, if you like, laws of memory, if one wants to call it that, is the degree to which that you form a rich, elaborative association between the different elements that you're trying to remember, the better your memory will be. So, for example, if you see an object and a scene, and you imagine that object moving around in that scene, that's actually the instruction we give people. Memory for that pairing is way better than if I show you that object and I show you and the scene, and I just tell you and I ask you to do something like just classify the object as animate or inanimate. Absolutely. Generally speaking, you get much better memory when you don't have to be trying to remember, but when you engage in mental activity that means that the images of these items get blended together in your imagination.
Okay. What are you doing or saying to demonstrate selectivity?
Say that again, please.
What are you doing or saying to demonstrate?
So selectivity in our hands is the degree to which a brain region responds more precisely or more specifically to one class of objects than to another. So our measures of selectivity are the degree to which for example, the parahippocampal place area shows more similar patterns of activity to different scenes than it does to a scene versus a face or a scene versus an object. So the selectivity measure is kind of built into the way that we analyze the brain activity. I don't know if that answers the question, but we do it in two different ways for what it's worth. We have two different measures of selectivity, which rely on different underlying assumptions. And we get convergent evidence from both of these two different measures of selectivity. Nothing hangs on our cherry picking just one measure.
Mhm. Okay. Since priming memory remains relatively stable across age, would practice or use of strategies be helpful to enhance the working memory?
So working memory is a different animal, unfortunately. So working memory is impacted. I didn't mention it today because I was only talking about what we would refer to as long-term memory. Working memory is the ability not just to hold information in mind, which we're very good at as we get older, but it's the ability to manipulate that information and to use it in a strategic way. So for example, simple short-term memory would be repeating back to me a string of digits that I read out to you. Working memory would be repeating back to me a string of digits where you've added one onto each of the digits. We're great at the short-term memory bit. The working memory bit declines as we get older. Things like priming, an example of something that would be under the rubric of priming would be a simple motor skill. For example, learning, I don't know, to use, this sounds stupid, but learning to use a new kind of kitchen implement, something that I'm abysmal at. You get better at it with practice. That kind of thing, that kind of experience-based, practice-based learning, which requires memory. It's memory. If you didn't have a record of the prior experience, you could not benefit from practice. That kind of slightly automatic kind of learning is not impacted very much by aging at all.
Okay. Another question, why is onset getting younger? Is it because we have better medical knowledge and testing?
Sorry, I didn't hear the first bit of the question.
Why is early onset diagnosis, early onset dementia?
So early onset Alzheimer's is considered by many people to be possibly even a different disease. So for most people with early onset Alzheimer's, people who develop the disease in their 40s or 50s, the overriding majority of those individuals have one of three different genetic mutations. That means that they are almost certain to develop the disease. Early onset Alzheimer's was not really recognized. It used to be called pre-senile dementia. And it was not really recognized for a long time. And the ability to genotype and to identify people who carry the mutations is a relatively recent development. So early onset Alzheimer's is extremely important. It may give us crucial clues into late onset Alzheimer's, which is, by the way, more than 95% of people with Alzheimer's have late onset Alzheimer's. Early onset is very rare, but it just reflects the fact that the scientific and medical community really didn't, to be honest, wake up to the importance and significance of the disease until relatively recently.
Thank you for that. And Dr. Rugg, you have a few fans out here, so this is your big opportunity. Do you have a blog or social channel to follow your insights? There are several people out here today on the chat that thought this was a fascinating talk, and the way that you explained it was so accessible. And they're hoping that you have a blog or some way that they can follow you and your research.
Well, thank you. My lab has a Twitter site.
And you'd like to give it that?
It is, I don't do Twitter, so, but it is, the Twitter handle is FNIM_lab, LAB, FNIM lab.
Okay, great.
And we also have a lab website, and CVL itself, CVL_neuro, has its own Twitter site as well. But I will ask in fact, our Twitter supremo in my lab is in fact Sabina Strakova, a very, very talented graduate student who did some of the work that I was describing today. She is in charge of our Twitter site, and I will tell her she needs to up the number of, increase the number of tweets.
Okay, great. So just so everybody knows right now, we're going to respond to that via the Q&A, and then we'll also put it out as well.
Okay, great. Thank you very much.
Okay. We're researching that right now for you. So that concludes our program. And Dr. Rugg, thank you again for taking the time to speak with us today.
It's been a pleasure.
And thank you all. Thank you. And thank you all for joining us for the Aging Mind Foundation Lecture Series webinar. If you wish to revisit this presentation, we will be posting a recording to our YouTube channel, which can be found by searching Aging Mind Foundation. Monday to YouTube and our website, agingmindfoundation.org. You can find all of this season's webinars on YouTube and our Aging Mind Foundation website. Thank you all for joining us, and have a great week. And please watch our website for other webinars as the year goes on. Thanks so much, everyone.